Let us take a look at the law or principle of independent assortment. This is a principle Gregor Mendel derived using a dihybrid cross. This dihybrid cross is a breeding experiment where a researcher will be following the inheritance of two characteristics simultaneously. In this example you see on the screen, you can see one of those classic experiments done by Gregor Mendel, where he wanted to figure out the results, the outcome of a cross between a plant that produced yellow and round seeds, as you can see here in this example, and another plant that produced green and wrinkled seeds. These are two true breeding parents, meaning that they have been selected from parents, as you look at this case on the left, that only produced round and yellow seeds, and the other parent was selected from parents and grandparents that had only produced wrinkled and green seeds. One of the details you always have to keep in mind when you're doing breeding or genetics experiments is that you have to carefully identify the genotypes of the parents or the P generation. So let's begin with this. We had learned previously, as we explored the law of segregation, that Mendel stated for each inherited characteristic, all organisms have two copies of a heritable factor. We call these genes now two copies of a gene, and one inherited from each parent. So you can see here for this yellow and round seed producing parent, there's going to be two capital R's and there's going to be two capital Y's. We can extend the same logic to the parent producing the green and wrinkled seeds. So for the shape of the seed, there will be two lowercase r's, which is the representation for the recessive wrinkled allele, and two capital Y's, which will be the symbol for the recessive green allele. And so now that we have identified the genotypes for the parents, the next step is going to be figure out the kinds of gametes these plants can produce. I'm going to be switching here to laser pointer mode so I don't make a mess out of this slide. So for this parent that is producing the round and yellow seeds, there's only one type of gamete that they can produce. When the capital R and the capital R get separated, we're still applying the law of segregation to figure out the gametes involved in this type of a cross. So as these two capital R's get separated, one goes into one gamete, the other one goes into the other gamete, the Y's get separated too, but in the end, capital R's only go with these capital Y's for this parent that is homozygous dominant. So there's only one type of gamete. There's no other combination of capital R's or capital Y's we can see in the formation of the gametes. Another detail you have to keep in mind here that remember is that the gametes only have half of the genetic information found in the parents because gametes are made by the process of meiosis. And meiosis always reduces the number of chromosomes or genetic material in half. For the parent with the green and wrinkled seeds, the same is going to happen. There's going to be only lowercase r and lowercase y, so they can only produce one type of gamete. When the cross is made between these two plants, all of the plants in the F1 generation are going to be inheriting a capital R, a lowercase r, capital Y, lowercase y. As you inspect the capitals and lowercase, as you can see that this capital R means that the seeds are going to be round because round is dominant to wrinkled. There is also a capital Y over here next to the lowercase y, but because the capital Y is dominant, and that's for yellow seeds, then the seeds are going to be yellow. So all of these plants in the F1 generation are going to look just like the homozygous dominant plant. The question is now, what would happen when we cross two of these plants from the F1 generation? You will be crossing here two of the same descendants from the two original parents. So you're crossing sibling plants. It's okay to do this with plants. We're going to be learning why this is not okay to do with animals. Then let's figure out the kinds of gametes that can be produced by a parent with this type of genotype. But before we do that, let us just assume for a moment, what if there was no independent assortment? And so we can develop a hypothesis here. As you can see in this illustration, let's develop a hypothesis that there is dependent assortment. That somehow in the formation of the gametes for these F1 individuals, the capital R's always find a way to connect with the capital Y. In that case, capital Y and capital R go together into one gamete, and the lowercase r and the lowercase y goes together into another gamete. And so the eggs produced will have either 2 dominant alleles or 2 recessive alleles, and the same for the plant responsible for producing the sperm. Now, again, I want to emphasize, why are we doing this in this situation? We are looking at the hypothesis of dependent assortment. And if you're thinking, depending on what? What depends on what? What if this cross depended on dominants always going together with dominant alleles and recessives always going with recessive alleles? If that is the case, the outcome of the phenotypes for the F2 generation is going to be similar to what we saw in the law of segregation. There's going to be 3-to-1 ratios. 3 of the F2 generation plants producing the dominant traits and 1 out of the 4 producing the recessive phenotype. But what we know is that this is not the case. And so this principle of independent assortment becomes a confirmation of what we learned previously in meiosis when we said that pairs of homologous chromosomes are going to be lining up independently from any other pair at the metaphase plate during metaphase 1. So if there is no dependence of dominant always going with dominant and recessive always going with recessive, if any allele can go with any other type of allele, as long as you have one of each type of characteristic represented by the alleles in the gamete, then here's what we can see instead. For a plant of the F1 generation, now we're looking at the hypothesis of independent assortment. What you can expect to see is that this capital R can go with a capital Y. We have that in this gamete right here. But capital R can also go with lowercase y because the assortment is independent. And so here we have this capital R with lowercase y. Lowercase r can go with capital Y. Yeah, we have that over here. And lowercase r can also go with lowercase y, and we have that represented in this situation. For the other for the other plant responsible for making gametes, the same 4 types of gametes can be found. And so when you do all the basic work of crossing an egg and a sperm, one of these eggs with one of these sperms, and then you come up with all of the phenotypic possibilities, you're going to find a phenotypic ratio of 9-to-3-to-3-to-1. And when you see this kind of a pattern, when you're crossing 2 heterozygote parents in a dihybrid cross, that is a signature that independent assortment has occurred and that alleles don't segregate in any pre-established pattern, that alleles are going to be segregating at random in an independent fashion from other pairs of alleles going into a gamete. What I want to do now is I want to help you with a worksheet that we have previously you had received previously in class, and I'm trying to change my screen here to what that screen looks like. Okay, so here we go. And so at the previous class, I gave you a paper that looks like this one, and what you want to do is you want to take the time to carefully read. I'm not going to do that for you in this video. You want to read the scenario that is presented, but it's essentially an extension of the example I went over with you in the PowerPoint slide we have just covered. And so what you want to do, remember the strategy that I presented for you in class. What you want to do here is you want to figure out first the genotypes of the parents. So I want to be able to do some annotations. We'll see if this will allow me to. Okay, so get your worksheet, get it ready, pause this video if necessary, go get your worksheet, and then resume once you have your worksheet with you. In the previous description, we're told that brown is going to be a dominant characteristic, and that short hair is going to be a dominant characteristic. We're also told that white and long-haired are going to be recessive characteristics in this case. And so what are the genotypes of the parents? This is how you do it. For the genotypes, you have to remember that brown is dominant, and so we're going to put here 2 capital Bs. And so why 2 capital Bs? Remember that for the genotypes, all individuals have inherited 1 allele for each trait from each of the parents the organism had. Because brown is dominant, we're using capital B. Now, for short hair, I'm going to use the first letter of the word hair, H, and so I'm going to put here 2 capital H's. And I'm sorry, I'm going to try to redo this because it's kind of hard to write on these screens. So I'm going to do once again. I'm trying to just annotate. Okay, here we go. Capital B, that's a little bit better. Capital H, and capital H. So capital B is a symbol for brown, capital H is a symbol for short hair. For the rabbits that are producing white hair, we're going to be using lowercase b and lowercase h's for the short hair. So that's the genotypes of the P generation. Now, what are the gametes that these parents can produce? Since the brown short hair parent only has 1 type of brown and 1 type of length of the hair allele, the only kinds of eggs that this female rabbit, by the way, they call them does, a female rabbit is going to be called a doe. They can only make 1 type of gamete, and it's going to be the one that has the 2 dominant alleles. What about the male over here? Male rabbits are also known as bucks. And I'm telling you this because in another exercise, it talks about rabbits, and then it says about does and bucks. So now you know the meaning of those words too. So this one only has lowercase b and lowercase h alleles, so there's only 1 type of sperm this rabbit can make, lowercase b and lowercase h. So what would be the genotype of the F1 generation? Combine these 2 gametes, and then you end up with capital B, lowercase b, capital H, and lowercase h. That's the genotype of the F1 generation. Then you're asked, okay, so what would be the gametes produced by these kinds of rabbits on the F1 generation if we were to cross them, if we wanted to produce rabbits from this generation? So you have to have a male rabbit and a female rabbit. And so for the male rabbit, what kinds of sperm cells can you make? Apply the principle of segregation. Any kind of B can go with any kind of H. That's all it says. And so here we can have, for example, capital B with capital H. These 2 can go together. On another one, we can have capital B and lowercase h. That's also a possibility. Now let's take a look at the lowercase b. We can have lowercase b with capital H. Sure, why not? It's independent assortment. And we can also have lowercase b with lowercase h. 4 different kinds of combinations of b's and h's can be obtained. And if you don't believe me, you can do the try on a piece of scratch paper, and you will see that there's only 4 different combinations. How about for the female responsible for making eggs? The same kinds of combinations will be seen. Capital B, capital H, capital B, lowercase h, lowercase b, capital H, lowercase b, and lowercase h. And what you do now in this type of a cross is begin putting in each square where 2 gametes will be meeting. If this sperm here meets with this egg over here, then you will have capital B, capital B, capital H, capital H. And so you can take the time to do all of those crosses that you can combine. And at the end here, you will be writing the number of individuals of a specific phenotype that you will be seeing. And you will see that ratio once again of the 9 to 3 to 3 to 1 we saw previously with the yellow and brown seeds versus the green and wrinkled seeds. So I'll let you figure this out. I'll let you work on this, but I've given you the head start on how to solve this exercise on independent assortment.